This is the brief on kidney cells, stress, and the massive spaced effect. Okay, so get this. It turns out that cells outside your brain, yeah, even like your kidney cells, are incredibly sophisticated. And their outcomes, whether it's forming memories or, painful stones, it really relies entirely on the timing and repetition of the signals they get. First, let's look at the negative side. What happens with constant repetition, like in stress-induced kidney issues? See, chronic stress keeps triggering hormones like cortisol and vasopressin. It's this constant flood, not just a quick burst, that harms the kidneys. It boosts calcium in urine, cuts down urine volume, makes it super concentrated. Basically the perfect setup for kidney stones. Second, though, there's this really fascinating positive side called the massive spaced effect. It's pretty simple. Signals given with breaks, spaced out, work better than one long continuous blast. Some groundbreaking research out of NYU showed that non-neural human cells, yeah, kidney and nerve tissues included, actually show memory-like behavior. When they got chemical signals in spaced pulses, bam, they activated a memory gene and had a much stronger, longer-lasting response. Finally, here's how it connects. The timing pattern literally determines what happens to the cell. Think about it, the exact same kidney cell type responds completely differently. Chronic, non-stop stress signals, that leads to problems, pathology. But timed, spaced-out chemical signals, that actually leads to enhanced cellular memory. Ultimately, it shows that for cells throughout your body, how and when they receive input is absolutely crucial.
We often talk about memory, where it lives, how we store and retrieve information, and that conversation, it almost always starts and, frankly, ends with the brain.
Right. Brain gets all the credit.
Exactly. We tend to forget, or maybe just not consider, that memory, or perhaps a kind of cellular intelligence, could be a property of all our cells.
Yeah.
Even in organs you don't really associate with thinking, like the kidneys.
And that's the friction point we're diving into today. We've got these two, seemingly very different stacks of sources.
Okay.
On one hand, you have clinical documents, personal notes too, about kidney stones, specifically linking them, or seeming to link them, to chronic stress as a physical trigger.
Right, the physical manifestation of stress.
Yeah.
And on the other side.
Completely different field. Groundbreaking research from NYU, really, on molecular biology, stuff that challenges how we even think about biological memory. It introduces this concept of non-neural cellular memory. They call it the massive spaced effect.
Massive spaced effect. Okay, so kidney stones and stress over here, cutting-edge cell biology over there. How do they connect?
That's our mission for this deep dive, to show that these aren't separate things. They're governed by basically the same biological principle. Kidney cells are incredibly sensitive to the timing and the repetition of signals they receive.
So it's not just what the signal is, but how it arrives. Like, is it constant noise or a specific rhythm?
Precisely. Whether it's a chaotic flood of stress hormones day after day or, a carefully timed chemical pulse in a lab dish.
Yeah.
That pattern, that temporal pattern, seems to determine the outcome for the cell.
The cell is processing rhythm. Wow.
Yeah.
Okay, let's unpack this, starting with the human cost. Let's get into the reality of kidney stones first.
Right.
For anyone listening who hasn't, thankfully, experienced a kidney stone, it's probably easy to gloss over the whole urinary tract system, but we need to quickly map it out to understand the pain.
Sure. So you've got the kidneys, obviously doing the filtering. Then the urine travels down tubes called ureters to the bladder, which stores it. And finally, out through the urethra.
Simple enough, but you mentioned a choke point.
That's the ureter. It's the absolute key here. The kidney itself, where the urine collects initially the renal pelvis, it's relatively wide, kind of like a funnel.
Okay.
But then it narrows dramatically into this thin tube, the ureter, which is often only, a few millimeters across.
Ah, so when a stone forms and starts to move.
It hits that bottleneck. It's literally like trying to shove a pebble down a straw. The blockage itself and then the body's reaction to it, that's where the agony comes from.
And this isn't some rare condition, right? The risk seems surprisingly high.
Oh, definitely. The recurrence risk is significant. Clinically, the data suggests that if you've had one stone, your chance of getting another one within 10 years is about 50%. 50%. That is, if you don't make some real lifestyle changes.
And family history plays a role, too.
Absolutely. If kidney stones run in your family, your personal risk goes up quite a bit. Genetics are definitely a factor.
The sources also flag specific groups beyond just, say, diet or dehydration. Who else is particularly at risk?
Well, pregnancy is one. Hormonal shifts, plus the physical pressure on the urinary tract, can slow down urine flow, creating conditions for stones.
Excellent.
Then there's low mobility. People who use wheelchairs or are bedridden for long periods. The lack of movement actually changes how your body handles calcium and other minerals, increasing the risk.
Interesting. And what about underlying diseases?
Yes, that's crucial. Things like inflammatory bowel diseases, Crohn's disease, or ulcerative colitis, they really mess with how your gut absorbs nutrients and minerals. Sometimes you absorb way too much oxalate, which is a major stone component.
Oxalate. We'll come back to that, I'm sure.
We will. And also, certain inherited metabolic disorders can basically program your body to produce specific kinds of stones, like cystine stones. They have their own unique challenges.
Okay, let's talk about the pain itself. It's called renal colic. And the sources say it comes in attacks or waves. Why isn't it just constant grinding pain?
That wave pattern is really characteristic. It tells you what's happening mechanically. When that stone gets lodged in the narrow ureter, the muscle in the ureter wall doesn't just sit there. It starts these powerful rhythmic contractions. It's called peristalsis.
Like the intestines moving food.
Exactly the same principle. The muscle is trying desperately to squeeze that stone downwards to push it toward the bladder.
So the body's attempt to fix the problem is actually causing the intense pain waves.
Precisely. The pain skyrockets during that muscle squeeze, then it might ease off, just a little, as the muscle fatigues momentarily. But then it recovers and clamps down again. That's the wave.
And there's that other symptom mentioned, Bewegungsdrang, this intense restlessness. People can't sit still.
Yeah, it's quite distinctive. You see patients pacing, writhing, constantly shifting position, trying anything to find relief. But unlike, say, appendicitis pain, where moving makes it worse, here the movement doesn't actually help the stone pass or stop the muscle spasm.
So why the urge to move?
It seems to be just this primal response to the overwhelming internal sensation. The body's just desperate to escape the feeling, even though changing position doesn't fix the underlying mechanical issue.
Okay. So someone experiences this, maybe they pass the stone or they need medical help. The sources are really clear. Collecting the stone for analysis is vital. Why?
Because that analysis is everything for prevention. Is it mostly calcium oxalate, uric acid, struvite, which is infection-related, or cystine? The composition dictates the entire strategy going forward.
You can't treat them all the same way.
Not at all. Advice for preventing a uric acid stone might actually increase your risk for a calcium oxalate one. Knowing the stone type tells you exactly what dietary changes, what medications might actually work. It's about tailoring the approach.
Makes sense. So how do doctors figure this out initially and check for damage? What's the diagnostic process look like?
Usually starts with urine analysis, simple dipstick test, maybe microscopy. They're looking for obvious things like blood hematuria. A stone scraping the lining often causes bleeding.
Okay.
They also look for white blood cells, leukocytes. That could signal an infection piggybacking on the stone, which is dangerous.
And the pH, the acidity you mentioned, that's important.
Hugely important. Urine pH gives a big clues. Very acidic urine, a low pH, points towards uric acid stones. Highly alkaline urine, high pH, might suggest those struvite stones linked to certain bacterial infections. It helps narrow it down right away.
Then comes imaging, usually a CT scan. The sources mentioned using it with and without contrast dye.
Right. For just finding the stone, you usually don't need contrast. Stones are dense, mineral things. They show up really clearly on a standard CT scan. Quick, easy, it tells you location, size.
So when would they inject the contrast dye?
The contrast is used when they need to see the plumbing itself, the soft tissues, and critically, the flow of urine. You inject the dye intravenously, it gets filtered by the kidneys, and then lights up the ureters and bladder on the scan.
So you can see if there's a blockage.
Exactly. Is the urine flowing past the stone, or is it completely blocked? A total blockage, what they call Avlode belemmerung, can back up urine and damage the kidney pretty quickly if it's not relieved. So the contrast shows the functional impact, not just the stone's presence. It's vital for deciding if urgent intervention, like surgery, is needed.
Okay, we've got the physical picture, the anatomy, the pain, the diagnosis. Now let's pivot to that trigger connection, the link between chronic stress and actual kidney damage. This comes partly from those personal sources, Dennis Freya's notes, describing this pattern and even like a physical weak spot feeling near the kidneys after stressful events.
Yeah, and what's fascinating here is how does something psychological like stress or a sudden shock translate into hard physical pathology like a kidney stone? It often gets dismissed as just anecdotal.
Right, it's all in your head.
But the sources actually lay out a specific biological pathway. It's not vague. There's a mechanism.
Let's get into that mechanism.
Yeah.
What specific hormones are we talking about when we say stress hormones? And how do they literally encourage crystals to form?
We're primarily looking at the big players in the stress response. Cortisol, that's the main long-term one. Adrenaline or epinephrine for the immediate jolt. And critically for this story, vasopressin.
Vasopressin. Isn't that also called ADH, antidiuretic hormone?
Exactly the same thing, ADH. And it does what the name suggests. It works against diuresis, meaning it reduces urine production. When you're chronically stressed or have that sudden fright, your HPA axis, the hypothalamic pituitary adrenal system, kicks into gear and releases these hormones.
And this hormonal flood creates, as we noted, this trio of harm for the kidneys. Let's break that down. How does ADH concentrate the urine?
This is central to the whole timing aspect. Think about fight or flight. Your body wants to conserve resources, especially water, to maintain blood pressure if you get injured. Vasopressin, ADH, directly targets the final parts of the kidney tubules, the collecting ducts.
Okay, what does it do there?
It makes the walls of those ducts much more permeable to water. It does this by triggering the insertion of special water channels, they're called aquaporins, into the cell membranes.
Like opening little gates for water to leave the urine.
Precisely. So loads more water gets reabsorbed back into your bloodstream instead of staying in the urine heading to the bladder. And that leads directly to the first harmful effect, a significant decrease in overall urine volume.
Less water. Got it. What about the second part, the minerals? You mentioned cortisol and calcium.
Yeah, cortisol's effect is a bit complex, but under prolonged stress, it can contribute to increased bone resorption. That's basically bone breakdown.
Stress makes your body break down bone.
It can, yeah. And when bone breaks down, it releases calcium into your blood. Your kidneys then have to deal with that extra calcium, trying to filter it out. So the second harmful mechanism kicks in. You get increased levels of calcium in your urine.
Okay, so less water leaving and more calcium being dumped into that smaller amount of water. I see where this is going.
Exactly. That's the third piece. You create highly concentrated urine with way more minerals floating around in it. The chemical environment inside the kidney becomes supersaturated.
Supersaturated, meaning there's more dissolved stuff than the liquid can normally hold.
Perfect analogy. And when it's supersaturated, basic physics takes over. It becomes much, much easier for those dissolved minerals, calcium, oxalate, uric acid, whatever, to start crystallizing out, clumping together, and growing into stones. Chronic stress basically sets the stage perfectly for crystals to form.
And this brings us back to that crucial temporal idea. A single stressful event, the body can probably handle it, flush things out, but it's the chronic nature, the repetition, that's the real problem.
That's the key point the sources emphasize. If that stress signal is hammering the system constantly, day in, day out, then ADH levels stay high, those aquaporins stay open, water keeps getting pulled back, and the urine stays concentrated. That supersaturated state isn't temporary, it becomes the norm.
So that weak spot feeling someone might describe.
It could well be the subjective sense of this relentless physiological pressure, the kidneys being forced to work in this highly inefficient concentration maximizing mode constantly. It's under sustained biochemical assault.
And stress rarely impacts just one system in isolation. Right. Let's talk about this gut-kidney stress axis. How does stress mess things up elsewhere in a way that affects the kidneys?
Oh, absolutely. Chronic stress is known to wreak havoc on your gut microbiome. It often leads to dysbiosis, basically, an imbalance where the bad bacteria take over and the good ones decline.
Okay, gut bacteria. How does that connect to kidney stones? Seems like a leap.
It's actually quite direct, especially for the most common type, calcium oxalate stones. There's a specific beneficial bacterium, Oxalobacter formigenes, whose entire job essentially is to eat oxalate in your gut.
It degrades oxalate from your food.
Exactly. It prevents it from being absorbed into your bloodstream. But when stress causes dysbiosis and oxalobacter levels drop, suddenly your body absorbs much more oxalate from your diet.
Wow. So stress hits the kidneys directly by concentrating urine via hormones, and it hits them indirectly by messing up the gut, so more stone-forming material gets absorbed.
That's the double whammy. You've got more oxalate coming in from the gut, putting extra load on the kidneys. At the exact same time, the kidneys are being hormonally primed to concentrate everything. It creates this vicious cycle. Inflammation, high oxalate load, high concentration. Perfect storm for stone.
Which brings us to that slightly cruel irony mentioned in the personal notes, the T connection.
Yeah, it's such a perfect little example of this mind-body tension. The notes suggest intellectually, the act of making and drinking tea can be about curiosity, connection, maybe finding a moment of calm, right?
Yes.
A stress reliever.
Sure.
But biologically, many popular teas, especially black tea, and definitely typical iced tea preparations, are incredibly high in oxalates.
Oh, no. So the very thing someone might reach for to mentally cope with stress could be physically increasing their risk if they're susceptible.
Precisely. The ritual meant for comfort becomes a biological risk factor. It just highlights that conflict perfectly and sets us up nicely for the next part, looking at how cells can respond positively to patterns instead of just negatively to chaos.
Right. So we've seen how chronic, maybe chaotic, negative signals like relentless stress hormones seem to push kidney cells towards disease. Now we flip the script using that NYU research. How can precise, timed repetition of a signal actually program cells towards something like intelligence? This really forces us to rethink memory, doesn't it?
It really does. This research, led by Dr. Nikolai Kukushkin at NYU, it directly challenges that old neuroscience dogma that memory is only in the brain, all about synapses. They're proposing that the basic machinery for memory might be much more fundamental, much older, present in many cell types.
And this leads to the massive spaced effect. I know that term from studying for exams, spacing out your study sessions works better than cramming all night. How on earth do you test that on a kidney cell?
Well, the hypothesis started with genetics and molecular pathways. They thought maybe the fundamental molecules and signaling cascades that neurons use for long-term memory formation, things involved in synaptic plasticity, maybe those same pathways exist and are functional in other cells too.
So they looked for the same molecular machinery.
Essentially, yes. And the core idea of massive spaced holds. Information seems to stick better if the input signal comes in chunks with breaks in between spaced rather than all at once massed. They wanted to see if that timing principle applied at the cellular level, outside the brain.
Okay, the experiment itself sounds pretty cool. What cells did they actually use? And why is that important for our connection here?
This is the crucial link back. They cultured various human cells, including, specifically, kidney tissue cells. They also used nerve tissue cells for comparison, but the inclusion of kidney cells is the direct bridge.
So the very same type of cell that seems vulnerable to chronic stress signals
is the exact type they showed could exhibit this memory-like behavior in response to pattern signals.
Okay, but how do you know if a kidney cell remembers something? You can't ask it questions.
No. They used some clever molecular tools. They engineered the cells to have a reporter gene. This gene was designed so that when a specific memory pathway inside the cell got activated, likely involving key proteins like CREB, which is famous in neuronal memory.
Okay.
When that pathway fired up, the reporter gene would turn on and produce a fluorescent protein. Basically, the cell would start to glow.
So the glow is the readout. Brighter glow, longer glow means stronger memory.
Exactly. It gave them a visible, quantifiable way to measure how strongly and for how long that memory pathway was activated in response to a stimulus.
What was the stimulus? What chemical signals did they use?
They used chemicals known to trigger these intracellular signaling cascades involved in memory and plasticity in neurons, things like forskolin and 4-bol ester, PMA. These effectively mimic the kinds of signals a cell might receive naturally.
Right. Now the key test. Masked versus spaced. How do they actually deliver these chemicals?
They had two main groups. The masked group got a single, long, continuous dose of the chemicals over, say, a few hours.
All at once, like cramming.
Right. The spaced group got the exact same total amount of chemical, delivered over the same total time period, but it was broken up into several short pulses with rest periods in between.
Short bursts with recovery time. Okay. Here's where it gets really interesting. What happened? Which pattern did the kidney cells respond to better?
The results were apparently really clear, and they mirrored exactly what happens in learning and memory studies. The cells that received the spaced pulses showed a significantly stronger and much longer-lasting response.
They glowed brighter and for longer.
Yep. The activation of that internal memory gene pathway was far more robust and persistent compared to the cells that got the continuous masked signal.
That's fascinating. So the pattern of the signal, the timing, was everything. Why? What's the thinking on why spacing works better at a molecular level? What's happening during those breaks?
The leading idea is that it's about resource management and consolidation within the cell. Think about it. If the signal is just constant hammering away,
the cell gets overwhelmed.
Kind of. The receptors on the cell surface that detect the signal, the enzymes inside that relay the message, they can get saturated, or desensitized. They effectively burn out or stop listening if the signal never stops. It's like cellular fatigue.
Like trying to listen to loud static forever. You just tune it out.
Exactly. But with space signals, that short pulse activates the pathway. Then the break, the pause, gives the cell time to reset. Receptors can recover their sensitivity. The cell can synthesize new proteins needed to stabilize the change. It can sort of consolidate the effect of the first pulse before the next one hits.
So the rhythm allows the cell to actually process and integrate the signal properly, leading to a lasting change.
That seems to be the gist of it. The spacing allows the molecular machinery to work optimally, to transition from just a brief chemical flicker to a more stable, long-term change in gene expression, which is essentially what cellular memory is. The rhythm programs the cell for a more intelligent, lasting response. So yeah, we've kind of reached the core synthesis here. We started with the physical pain, the pathology linked to chronic, maybe chaotic, stress signals hitting the kidney, and now we see research showing those very same kidney cells possess this sophisticated ability to process timed rhythmic signals in a way that enhances their function, their memory.
The absolute key takeaway seems to be that shared biology. The kidney cell isn't just a passive filter bag. The fact that the same cell type is involved in both the stress-induced problems and this advanced cellular memory effect. It tells us something fundamental about the kidney, doesn't it?
It really does. It positions the kidney cell as a complex processing unit, highly attuned to temporal patterns. If we connect this to the bigger picture, its health, its function, seems deeply tied to how it interprets the rhythm of the signals it receives, not just the chemical content.
Okay, let's put the two findings side by side, using this idea of sensitivity to timing. First, the negative pattern, continuous chronic repetition.
Right. In the stress scenario, the kidney cells are getting bombarded by hormones, maybe erratically, maybe constantly. There's no helpful rhythm, no recovery. That continuous, overwhelming input leads straight to pathology.
The cell can't cope, can't recover, can't flush properly.
Exactly. Receptors might downregulate. The internal environment stays permanently optimized for concentration and mineral precipitation. The cell is basically programmed for disease by that relentless, chaotic signal. Stone formation becomes almost inevitable under those conditions.
Okay, now contrast that with the positive pattern, spaced, timed repetition.
In the lab experiment, those same cells, given the same total stimulus but delivered in that rhythmic, spaced pattern, they respond completely differently. They show enhanced function, stronger signaling, longer-lasting activation of beneficial pathways. They learn.
They're programmed for, like, resilience or optimized function by the rhythm.
That's a great way to put it. The spaced input allows the cell to respond intelligently, to adapt and strengthen its response over time.
So the conclusion feels almost stark. The temporal pattern of the input, whether it's internal stress hormones or external lab chemicals, seems to be the deciding factor. It dictates whether the cell goes down a path of disease or a path of enhanced function, maybe even cellular intelligence.
It really reframes how we should think about cellular health. It's not just about avoiding toxins or getting enough nutrients, which are obviously important. It's also about the rhythm of the signals our cells experience day to day. Timing is biology.
Okay, if kidney cells are this sensitive to timing and patterns, then all that standard advice about preventing kidney stones, it suddenly looks different. It's not just a list of rules, it's like a practical guide to giving your kidney cells the right rhythmic signals.
That's a fantastic way to frame it, because fundamentally, all stone prevention strategies are about interrupting that negative pattern, that masked state of supersaturation where minerals constantly want to crystallize.
So how do we provide the positive pattern?
The number one way is through consistent, adequate hydration. We're trying to provide a continuous, gentle, spaced input of water to keep the urine dilute.
What's the actual target? How much fluid are we talking about?
The clinical benchmark isn't just how much you drink, but how much urine you produce. The goal is to generate 2.5 liters of light-colored, clear urine every single day. And crucially, that volume needs to be spread out over the full 24 hours.
2.5 liters of output. That means drinking considerably more, right? Since we lose water through sweat and breathing.
Absolutely. Especially, as the sources note, when you're losing extra water elsewhere, hot weather, intense exercise, maybe you have a fever or diarrhea. In those situations, you have to consciously increase your fluid intake, that spaced input, to compensate and keep that urine output up and dilute. You're actively preventing the system from reverting to that risky, concentrated state.
So volume and timing are key. What about the chemistry of the urine? Can we provide positive chemical signals too?
Yes. The single most important natural inhibitor of calcium stone formation is citrate, which comes from citric acid.
Citrate, how does that work?
It works by binding to calcium in the urine. If citrate binds the calcium, then the calcium can't bind with oxalate to form calcium oxalate crystals, which are, 70–75% of all stones. So getting enough citrate is like sending a constant don't crystallize signal.
And the best natural sources are?
Citrus fruits are king here. Lemons, limes, oranges, grapefruits. Squeezing fresh lemon or lime juice into your water throughout the day is a really effective way to keep citrate levels up consistently. Again, it's that steady, spaced input.
Okay. Now, what about things to limit? The sources mentioned general restrictions for most stone types.
Two big ones are salt and animal protein. Too much sodium chloride, salt, makes your kidneys excrete more calcium into the urine. It directly promotes the conditions for calcium stones.
So it mimics one of the negative effects of stress hormones.
In a way, yes. The advice is usually to limit salt to around 6 grams per day, which is tough because it's hidden in so many processed foods. You really have to be mindful.
And animal protein. Why limit meat?
High intake of animal protein increases the overall acid load on your body. To buffer that acid, your body might break down bone slightly, releasing calcium, and your kidneys also tend to excrete less citrate when the acid load is high. So it's a double negative, more calcium, less inhibitor. The general advice is often no more than about 100 grams, maybe 3 to 4 ounces of meat per day. Keep the system less acidic, less loaded.
Makes sense. Now, the advice gets even more specific depending on the stone type. Calcium oxalate stones, the most common, need limits on high oxalate foods.
Right. Things like spinach, rhubarb, beets, nuts, chocolate, strong tea, especially iced tea. You don't necessarily have to eliminate them, but moderation is key. Interestingly, dairy is usually okay in normal amounts for calcium oxalate stones, despite the calcium in food can actually help by binding oxalate in the gut. And citrate is protective.
But then there are those cysteine stones. The sources highlighted them as needing maximal effort, really emphasizing timed intervention.
Yes, cysteine stones are tough. They're genetic, recur frequently, and the only way to prevent them is extreme dilution. Patients need to produce a huge amount of urine, like 3 to 4 liters per day, consistently.
3 to 4 liters, how do you even manage that, especially overnight?
That's the ultimate test of this timing principle. Because urine naturally concentrates overnight while you sleep, cysteine stone formers often have to maintain high fluid intake around the clock. The sources explicitly mention the need to set an alarm clock to wake up in the middle of the night, specifically to drink water.
Wow. You have to force the rhythmic input.
Exactly. You need to produce maybe 1.5 liters of urine, even during the sleeping hours, to prevent that cysteine from reaching saturation point. It's a massive commitment, but it directly illustrates the core idea. You overcome a continuous biological problem, high cysteine excretion, with a rigorously timed, continuous preventative input, water. It turns drinking water into a precisely scheduled medical therapy based on cellular timing. #outro.
This has been quite the journey. We started down in the weeds of kidney stone pain, that visceral experience linked in Dennis Verhage's notes to stress. And we've ended up seeing kidney cells, not just as filters, but as these incredibly sophisticated little computers, essentially, reading the rhythm of signals.
Yeah, differentiating between the biochemical noise of chronic stress, that continuous, potentially harmful pattern, and the structured, meaningful pattern of space signals that seem to promote better function, better memory.
It really drives home that the kidney, and likely many other tissues, are part of this body-wide temporal network, constantly interpreting timing. Chaos leads to breakdown, rhythm can lead to resilience.
Right, it's a powerful reminder.
So what does this all mean? If our kidney cells can be programmed towards disease by the timing of chronic stress, or towards enhanced function by deliberately spaced inputs, what does that imply about the broader patterns in our lives?
That's the big question, isn't it?
Think about the rhythm, or lack thereof, in your sleep cycles. Your meal timing, is it erratic or regular? How consistently do you manage emotional responses? Are these patterns potentially programming all the cells in your body right now, for better or worse?
It suggests that striving for a stable, rhythmic lifestyle isn't just about feeling psychologically better. It might be fundamental to operating our cellular biology correctly. Maybe timing is biology at a very deep level. Definitely something to mull over.
Indeed. Thanks for diving deep with us today. We'll be back soon with another exploration.
Welcome to the debate. Our source material today presents us with a really fascinating intellectual collision, I think. It draws a line between something deeply personal, Dennis Verhage's documented struggles with stress-induced kidney stone formation, and something very different, which is this groundbreaking scientific research into the massive spaced effect observed in non-neural cells.
It is a provocative convergence, certainly. And what seems to tie these two, quite disparate worlds together is the shared tissue, kidney cells. And the crucial observation, as the material notes, is that both processes, the, let's say, pathological one of stone formation and the experimental memory retention, are described as being highly sensitive to the timing and repetition of signals.
Right, exactly. And that shared sensitivity, that's what leads us straight to our core question today. Does this temporal coherence point towards a single, maybe unifying principle of cellular memory or even intelligence governing how kidney tissue responds? Or is this just a compelling but ultimately coincidental overlap of two fundamentally different biological pathways, one being, pathological and hormonal, the other informational and much more localized? I'll be arguing that these temporal dynamics are essentially the unified language of kidney cell response.
And, well, I'll be maintaining that we risk a kind of intellectual overreach here if we conflate what is, frankly, a catastrophic metabolic failure with a very specific type of localized cellular information storage. I'll be emphasizing the distinct nature of the underlying biological machinery and, frankly, the vast difference in scale between these two phenomena.
Okay. Well, my position really rests on the discovery of this massive spaced effect, particularly in kidney cells. This finding fundamentally challenges that old sort of neurocentric view of memory, doesn't it? It shows that kidney cells, which are non-neural, they actually possess the sophisticated molecular machinery needed to detect and leverage the interval between inputs. This suggests a fundamental property of what we might call cellular intelligence. Repetition and timing matter. They matter deeply. So in the experiments, brief, spaced chemical pulses lead to powerful, long-lasting gene activation, which looks an awful lot like true cellular memory.
Okay, but how does that specific controlled experiment relate to, say, a patient like Verhage enduring chronic, messy, real-world stress?
Well, it establishes the template, the framework for temporal analysis by the cell. So now, if we apply that template to Verhage's experience, well, chronic, repeated stress translates directly into a continuous, essentially masked negative signal. This comes primarily from those persistent stress hormones like cortisol, vasopressin. The cell, under this kind of bombardment, it seems unable to reset or differentiate these continuous inputs effectively, and that leads to system breakdown. You get the concentrated urine, the high calcium levels. The pattern dictates the outcome. Spaced, pulsed signals seem to build functional memory. Chronic, masked signals seem to build structural pathology. Now, I see why you think the functional outcomes look so different, destruction versus creation. But let me give you a different perspective. Maybe the temporal pattern itself, whether it's masked or spaced, is the unifying biological variable here. It determines whether the cell responds constructively or pathologically.
Well, that framework, that idea of pathological memory, is intellectually neat, I suppose. I find it fundamentally fails to account for the biological reality of the stone formation mechanism. The core issue for me here is really the difference in scale and function. Look, the mechanism behind kidney stones, it's a complex systemic pathology. It's driven by the endocrine system. Stress hormones flood the body, right? And they directly signal the kidneys to drastically alter fluid dynamics, decreasing urine volume, increasing mineral excretion. This is immediate. It's acute metabolic dysregulation. It's a whole system response to what the body perceives as a threat.
A response, though, that is sustained through repetition, wouldn't you say?
Sustained, yes, absolutely sustained. But is it necessarily remembered in the same informational sense as the spaced effect experiment? I'm not so sure. The massive spaced effect, conversely, that's an isolated in vitro investigation. It's looking into sophisticated information retention. The memory effect there is triggered by very specific pharmacological activators, forskolin and formyl ester, things that manipulate localized PK and PKC pathways to force a genomic change. So, one is systemic pathology driven by endocrine signaling and fluid dynamics. The other is a localized, targeted observation of cellular learning. We really cannot conflate pathological hormonal damage with enhanced cellular information processing based solely on signal repetition. I'm sorry, but I just don't buy that. Let me tell you why. The functional gulf between, say, altering systemic urine pH and mineral concentration and successfully activating a specific PCAP, PKC-driven memory gene, well, it's just too vast, I think, to claim a single unified mechanism of intelligence is at play.
Okay, let's focus on that functional difference you raised, that distinction between destruction and creation. You emphasize rightly that stress hormones cause negative physical manifestations, what the source calls a harmful mechanism. But if the cell possesses this capacity for sophisticated temporal analysis, which the massive spaced effect suggests it does, why can't we interpret chronic stress pathology as the cell essentially, well, learning to fail? The cell is processing the repeated stress signal, right? And it's consistently shifting its fluid regulation into a high-risk state. It's a kind of long-term commitment to pathology, and it's driven by that repetitive input.
But that interpretation, I feel it minimizes the sheer physical and chemical burden placed on the kidney during chronic stress. We know this pathway involves the complex gut-kidney stress axis. So, chronic stress can cause gut dysbiosis, which then generates toxins, increases inflammatory markers. The kidney isn't simply registering a time interval like in the experiment, it's being genuinely overwhelmed by a flood of hormones and metabolic waste products. This is really a multi-system failure involving inflammation, metabolic alterations. It's a physical, structural compromise. A systemic pathology of this magnitude, it's fundamentally operating at a completely different level of biological organization than, say, a culture dish experiment activating a single reporter gene with a couple of chemicals.
I absolutely acknowledge the immense complexity and the systemic nature of the stress axis. I don't dispute that. However, complexity doesn't necessarily negate the underlying cellular intelligence. Perhaps it harnesses it or maybe overwhelms it in this case. The source material detailing Verhage's experience, it clearly emphasizes that the maintenance of those harmful stone-forming conditions is directly tied to the repetition of the stress state. And the kidney cells are the final common tissue where this failure manifests, aren't they? They are constantly being signaled, maintain this state of concentrated fluid. So, if we accept that kidney cells can achieve constructive long-term informational storage via temporal patterns, why is it such a stretch to think they could also commit to a destructive long-term functional state pathology based on a different, specifically masked temporal pattern?
Because the mechanism of that destructive commitment, that sustained high mineral concentration, it seems largely physicochemical. It's driven by things like high vasopressin, causing water retention, high cortisol, increasing mineral excretion. Its fluid dynamics and solubility limits essentially failing under pressure. To claim that the cell is performing sophisticated temporal analysis on these hormones when it might simply be responding to an elevated chemical load, well, I think that's stretching the definition of cellular intelligence maybe a bit too far beyond the current evidence.
That's an interesting point you make about pathological rhythm, but I suppose I would frame it differently. The temporal pattern of chronic stress isn't being read for informational change in the same way. It's simply sustained exposure that's overwhelming the normal homeostatic regulatory capacity.
And, to support my case for distinct mechanisms, we really have to drill down on the molecular difference between masked and spaced in that memory study. The mass-spaced effect uses chemicals that activate these specific pathways, PKA and PKC. Now, for the audience, these are like the cell's internal switchboards, right? The key to successful spacing seems to be that it allows critical newly activated proteins, specifically these catalytic subunits, it gives them time to actually move, to translocate from the cytoplasm into the nucleus and start writing that lasting instruction set into the DNA. That time gap is crucial.
Aha! But that molecular explanation is precisely what supports the unified view, in my opinion. It proves the non-neural cell has an inherent molecular clock, a temporal resolution capability that's optimized for signal intervals. The cell isn't just reacting chemically in a simple way, it's performing a complex timing operation to achieve genomic permanence. This temporal resolution capability, that's the common ground, I'd argue, regardless of whether the signal comes from a stress hormone or an experimental chemical. Now, let's just revisit the specific pathology you mentioned earlier. Even the resulting pain, the kidney stone colic, is described in the sources using that phrase in an anfallen kommt. It comes in attacks or waves.
Well, yes, but that's likely a symptom of mechanical disruption, isn't it? The stone moving or causing pulsed muscle spasms in the ureter. That's not necessarily the kidney cell's intelligence expressing itself. It's more likely the muscular system's reaction to a physical blockage.
Okay, but that pulsed pain is the severe manifestation of a process that's driven by chronic repetition over time. The stress state itself might not be perfectly smooth and constant. It's chronic and continuous, yes, but it's often likely pulsed in reality. Daily anxieties, repeated hormonal peaks and troughs throughout the day or week. This suggests the kidney tissue is constantly responding to this kind of pathological rhythm. If the cell's underlying function involves temporally resolving inputs via conserved pathways like PKA-PKC, then perhaps the chronic stress pathway is hijacking or exhausting this very same temporal mechanism, preventing the cell from properly resetting its fluid balance parameters. The pathological rhythm becomes the negative mirror image of the optimal spaced rhythm needed for positive adaptation.
I'm still not quite convinced by that line of reasoning, mainly because the core function being affected seems so different. It's regulatory versus informational. In the memory experiment, we are inducing a specific memory gene, right? A process aimed at long-term information storage. In chronic stress, the cells, particularly in the collecting duct, they seem to be simply failing to maintain adequate water reabsorption control because of the sustained elevated presence of hormones. You seem to be suggesting that the same sophisticated temporal resolution system that governs genomic memory is also being applied to manage immediate, minute-to-minute fluid regulation. That feels like a bit of a leap.
Well, I see it as applying the same fundamental cellular ability, the capacity to process time, to different outcomes. Why would the kidney possess such a powerful capability for temporal resolution only for a very specific set of experimental chemical inputs? It seems more likely that it's a fundamental, widespread property of the tissue, applicable perhaps universally to hormonal, chemical, and maybe even mechanical inputs. And if kidney tissue exhibits this sophisticated responsiveness, it fundamentally shifts our understanding of kidney function beyond just simple filtration, doesn't it?
Look, the implications of that NYU study are undeniably huge. I grant you that. Challenging the long-held view that memory is exclusive to neural systems, that's big news. But we really must exercise caution here. We are comparing data generated in a clean, highly controlled lab environment using brief, defined chemical pulses, comparing that to the chaotic, messy reality of a patient enduring chronic psychological and physiological stress that affects the entire gut-kidney stress axis. It's quite different.
But that external chaos is precisely why the internal temporal intelligence becomes so crucial potentially. If the cell's core language is temporal, then the timing of our therapeutic interventions becomes paramount. If chronic masked signaling causes failure, then maybe future strategies for kidney health should focus on introducing inputs, pharmacological or otherwise, in a carefully spaced, optimized manner, to allow the cellular repair and reset mechanisms the necessary time to actually engage. This suggests we need to look beyond just the concentration or chemical makeup of treatments and really address the timing of interventions.
I agree that considering timing for drug delivery is generally relevant, of course. But when we're tackling something specific like kidney stones, we have established evidence-based targets focused on chemistry and metabolism, things like decreasing urinary calcium, increasing fluid volume, managing pH. When tackling stones, I believe we should focus on the known chemistry and the clear metabolic failure, rather than suggesting that the kidney is somehow remembering stress in the same specific PKA-PKC-driven way it might remember a pulse of forskolin in a lab. The complexity here, I think, really requires us to appreciate the difference between metabolic failure and cellular information storage. They might both happen in the kidney, but that doesn't make them the same process.
Okay, so to summarize my perspective then, the source material, I believe, clearly demonstrates that kidney cells are highly sensitive to the temporal pattern of signals they receive. Whether this pattern leads to the negative outcome of chronic, masked, stress repetition, resulting in pathology like in Verhage's case, or the positive outcome of optimized signal spacing leading to robust cellular memory in experiments, the underlying sensitivity to the pattern of input seems to me to be a fundamental, potentially unifying aspect of kidney cell intelligence.
And I would summarize my view by saying that while the involvement of kidney cells in both phenomena and their shared general sensitivity to repetition is certainly striking, the evidence, as I read it, points strongly towards two distinct and operationally separate biological mechanisms. On one hand, hormonal metabolic dysregulation causing a physiological failure. On the other, a localized, informational, genomic process related to memory. The crucial lesson here, perhaps, is that complexity requires us to distinguish carefully between a coincidence of location and a true unification of molecular mechanism.
A critical distinction indeed, and one for our listeners to consider as they explore the material further. The profound influence that the timing and repetition of all signals, both internal, like stress hormones, and external stimuli, have on non-neural cells, particularly those in the kidney. Well, it certainly offers a rich area for continued inquiry.
Absolutely, and understanding whether this temporal sensitivity represents a singular universal language for these cells, or if it's merely a highly optimized trait deployed within different regulatory systems, well, figuring that out will likely shape how we approach medicine and biology for decades to come.